blueprint of life is the helical DNA molecule, which functions like a coiled zipper. In order to pass on the codes for the construction of protein, DNA unzips into two halves. The genetic code of the DNA molecule has to be read in order to be useful. The first step is the construction of RNA molecules along sections of the strand. Although RNA, or ribonucleic acid, is a long-chain molecule or polymer like DNA, it differs from DNA in several significant ways. Its sugar component, ribose, has one oxygen atom more than the deoxyribose of DNA. It is a single helix compared to the DNA double helix. RNA is smaller, but more plentiful. Like DNA, it is found in the nucleus, but also in the cytoplasm. While DNA may contain thymine, which bonds with adenine, RNA substitutes uracil when bonding with adenine during the process of RNA transcription. In addition, RNA is found in three distinct types: messenger RNA, transfer RNA, and ribosomal RNA, each of which serves a separate purpose in the process of protein synthesis. Protein synthesis begins with the transcription of messenger RNA along a section of the unwound and unzipped DNA strand. Once the complementary strand is completed, a protective tail, 200 nitrogenous bases long, is added, and the newly created messenger RNA passes from the nucleus into the cytoplasm, where it incorporates itself into the structure of one or more ribosomes. The outer membrane of the cell, in reality, is folded in upon itself in complex overlapping ways to form the endoplasmic reticulum, a vast canal system throughout the cell. The ribosomes are found lining this internal canal system. It is here at the ribosomes that amino acids are brought from the surrounding cytoplasm and assembled into linear strands of protein. The vital intermediary between the nucleic acid language carried from DNA by messenger RNA and the amino acid language of the working cell is the second of the three RNA types, transfer RNA. The various transfer RNA molecules act as vehicles for transferring amino acids from the cell's cytoplasm to the site of the protein synthesis in the ribosome. Transfer RNA is a small chain between 75 to 85 nucleotides long. It is folded back on itself so that the two ends of the chain are adjacent to each other and bent to form a rough L, with the two ends on the shorter arm of the L. Along the doubled over length of this chain, the nitrogenous bases are for the most part linked as in the DNA helix. Two exceptions, however, are important. At one end of the doubled over chain are three unlinked nitrogenous bases, which form a nucleotide triplet, or codon. This is called the acceptor end of the transfer RNA molecule. At the other end of the L is a loop in which the nitrogenous bases are also unlinked. This is known as the anticodon loop, and is the site where the transfer RNA molecule interacts with the messenger RNA strand. There exists a specific and unique transfer RNA molecule for each of the 20 amino acids which are involved in protein synthesis. An acceptor N which expresses the codon GCU, for example, codes only for the amino acid alanine. Similarly, transfer RNA with the acceptor codon AAG codes only for lysine, and GGC, in turn, only for glycine. In the translation of the genetic code from the language of nucleic acids to the language of amino acids, the first step is the binding of a free amino acid of the appropriate sort to the codon at the acceptor end of the transfer RNA molecule specific for the amino acid. Once the binding has occurred, the transfer RNA molecule ferries the amino acid to the protein assembly site, the ribosome. There, after incorporation into the ribosomal structure, the anticodon loop of the transfer RNA molecule interacts with the messenger RNA strand. The components of each anticodon loop fit only a specific codon on the RNA and form hydrogen bonds together. The ribosome then moves along the messenger RNA strand the distance of one codon. When the next transfer RNA arrives with its amino acid passenger, it locks into place in the same way. Now, the first amino acid is passed over and linked to the second through what is called the peptide bond. The first transfer RNA molecule is then released and becomes available to pick up another amino acid. Once more, the ribosome moves along the messenger RNA strand the distance of one codon to receive the third transfer RNA molecule with its amino acid passenger. Thus, the polypeptide chain of amino acids elongates until the synthesis of the specific protein is complete. This process, which churns out protein like ticker tape, can be fairly rapid, occurring once every second in a eukaryotic cell, and as many as 20 times per second in a bacterial cell. From a completed protein molecule such as insulin, which helps manage the sugar balance in the bloodstream and in the cells, we can trace a protein structure back to its DNA blueprint. The insulin molecule is known to consist of two polypeptide chains held together by sulfur bridges. Consider a small part of the amino acid sequence: glycine, isoleucine, valine, glutamic acid, and glutamic acid. The nucleotide codon for glycine is guanine, guanine, adenine. For isoleucine, adenine, uracil, and cytosine. For valine, guanine, uracil, adenine, and so on. This particular sequence of codons is created by complementary codons on DNA. Each of these letters, of course, represents a purine or pyrimidine-based nucleotide. And these, in turn, can have only one possible partner, allowing us to reconstruct the exact composition of the DNA molecule, which carries a portion of the insulin code. From the DNA blueprint, messenger RNA transcribes the genetic information and transports it to the ribosome, where it is translated by transfer RNA, which hauls free amino acids from the cytoplasm and links them in correct order. Ribosomal RNA, the last of the three RNA types, manages the interaction between messenger RNA and transfer RNA. The ribosome is a complicated two-section assembly line, which ensures that protein is correctly formed.